So, good morning. Let us revise biomolecule, that is nucleotide. Mainly, I am concentrating on Watson and Crick model of DNA, that is usually asked 10-mark essay. Okay, and a few MCQs or one word also asked in that. So, just an introduction, you know that DNA is basis of hereditary deoxyribonucleic acid. It carries genetic information from the from the parent to the daughter cells or to next generation by a process called replication. Okay, there are three processes: transcription, translation, and replication. Okay, so DNA is hereditary basis of life. DNA involved in protein synthesis. This is also one of the function of DNA. The genetic information in DNA is utilized for the synthesis of all proteins during the lifetime of cell and organism. So DNA also contains genetic code, so which is required for different types of RNAs, which is required for the synthesis of different types of RNA by a process called transcription, and RNAs are directly involved in protein synthesis. That is the function of RNA, protein synthesis in ribosomes. The process of protein synthesis is also called translation. Okay, so you know that what are nucleotides and what are nucleotides. There is a chance of short note. Nucleotide are building blocks of nucleic acid. What are those nucleic acid? Deoxyribose nucleic acid, DNA, and ribonucleic acid, that is RNA. So what it contains actually: nucleotide, nitrogenous base, pentose sugar, and phosphate group. So this nitrogenous base, purine and pyrimidine bases. Purine bases, adenine, guanine are major purine bases. Pyrimidine bases, cytosine, thymine, and uracil. Okay, so DNA contains pyrimidine base, thymine, but RNA contains pyrimidine base, uracil. Okay, that is the difference between DNA and RNA. Okay, that about the base. What about pentose sugar? Pentose means 5 carbon. In DNA it is deoxyribose sugar, 1 oxygen less at carbon 2. Okay, but in RNA it is ribose sugar, hence the name ribonucleic acid. DNA it is deoxyribose, means 1 oxygen less at carbon 2. There are phosphate molecules. Okay, one or 1, 2 or 3 phosphate molecules. So based on that they are named as monophosphate, diphosphate, or triphosphate. Best example is our energy rich adenosine triphosphate molecule, ATP. Okay, so nucleoside is nitrogenous base plus pentose sugar only. If phosphate group is attached to nucleoside, then it is called nucleotide. So nucleotide contains nitrogenous base, pentose sugar, that is ribose, and phosphate. Nucleotide is nucleoside plus phosphate. So there are many nucleotide, as I told: adenosine monophosphate, adenosine diphosphate, adenosine triphosphate, AMP, ADP, GMP, guanosine monophosphate, TMP, thymidine monophosphate, UMP, uridine monophosphate, etc. Okay, so other things not required. So as I told you, we are come to the structure of DNA, Watson and Crick model of DNA. Okay, Watson and Crick model of DNA. B form of DNA is double helix, described by Watson and Crick, is the most predominant form under physiological condition. Okay, uh, so here it is a 2 poly deoxyribonucleotide chains twisted around one another in a right-handed double helix. Okay, it is a right-handed double helix, so you have to underline right-handed double helix. Okay, so this double helix looks like a ladder, so you have to choose the right diagram, not the left one. Last time most of them drawn left diagram, choose right diagram. Okay, double helix, right-handed double helix twisted diagram. Okay, the sugar and phosphate part of the nucleotide, which makes the upright part of the ladder. Okay, that is sugar and phosphate, that is backbone of the helix. So which is backbone of right-handed double helix? Sugar and phosphate. So nitrogen bases of the nucleotide makes the rung of the ladder to climb, rungs of the ladder. Okay, these are the points you have to write. Okay, underline and write along with the labeled diagram. I am telling, write the twisted diagram, double helix. Okay, and label one, even nanometers, major groove size, minor groove size, internal diameter, that you have to label at least. Okay, and further uh, there are rules, base pairing rules, Chargaff's rules, what we call. Adenine always pair with thymine. Okay, adenine always pair with thymine, you can see 80, and guanine always pair with cytosine. So what is the bond between 80? That is double bond. Okay, but GC, cytosine, and guanine and cytosine, there is a triple bond. This maintains complementary a chain, complementarities of two strands, that is A always equal to T and C always equal to G. Okay, so these base pairs are held together by hydrogen bond. As I told you, GC 3 hydrogen bond and AT 2 hydrogen bond, and their two strands are anti-parallel. Two strands run in anti-parallel direction. You can see there, it is not crossing, it is anti-parallel. One strand in the 5 prime, see the top, one strand in the 5 prime to 3 prime direction, while the other strand is 3 prime to 5 prime direction. You can see the top and the bottom, that is slash is prime. Okay, 5 prime to 3 prime, 3 prime to 5 prime direction. The helix is 2 nanometer in diameter and makes a full spiral turn every 3.4 nanometer. One full spiral turn is 3.4 nanometer every after every 10 base pairs. Okay, one turn is there, that is 3.4 nanometer. So diameter of the helix is 2 nanometer. A full spiral turn uh, after every 10 base pair, that is the length is 3.4 nanometer. The distance between two base pair is 0.34 nanometer. Distance between two base pair is 0.34 nanometer. So there are one more point here to remember is protein interact with the DNA at the exposed bases present in two grooves. You know that there are two grooves, major groove 1.2 nanometer and minor groove. Okay, there are the major groove full turn and in between there is minor groove. So protein interact with the DNA at the exposed bases present in the grooves. There is a major groove 1.2 nanometer and minor groove is about 0.6 nanometer. These are parallel to the phosphodiester backbone, okay, and wind along the molecule. So this is about the silent features of double helix Watson and Crick model. So which is composed of two poly deoxyribonucleotide chain twisted around each other, okay, double helix. So it gives DNA a right-handed double helix structure, okay. And there is base pairing rule which is okay, and also there is double bond between AT, that is two hydrogen bond, and between GC there are three hydrogen bond, okay, three hydrogen bonds between GC and two hydrogen bond between adenine and thymine. As per base pairing rule, A always equal to T, C always equal to G, okay. So sugar and phosphate are part of the nucleotide. They are called as backbone of the helix, okay. So these are some of the points related to double helix model of DNA. RNA is a polymer of ribonucleotide linked together by 3' 5' phosphodiester linkage. I am not going much detail here. Today DNA I am taking. So difference between DNA contains millions of pair, RNA usually 100 to 5000 bases. See, DNA is double stranded but RNA is generally single stranded. So sugar present in DNA is deoxyribose but in RNA is ribose. Pyrimidine bases in DNA cytosine thymine but in RNA cytosine and uracil instead of thymine. In RNA uracil is present, okay. So DNA obeys base pairing rule, therefore A is equal to T and G is equal to C, but RNA does not obey base pairing rule. So A is not equal to uracil content and guanine content is not equal to cytosine content. So location, DNA mostly inside the nucleus and also in mitochondria, but RNA in cytoplasm. As far as the function is concerned, function of DNA is replication and transcription, but RNA mostly involved in protein synthesis, translation. So there are different types of RNA. I'm not going in detail. mRNA, messenger RNA, rRNA, ribosomal RNA, tRNA, transfer RNA, snRNA means small nuclear RNA, hnRNA is heterogeneous nuclear RNA. So functions of RNA, translation. So okay, so other details, so okay, not to bother, but you should be aware of the tRNA here. tRNA is transfer RNA. These are RNA molecules present in cytoplasm. Okay, okay. There are 20 species of tRNA corresponding to 20 amino acids present in the structure. They are called tRNA because they transfer amino acid from cytoplasm to protein synthesizing machine where ribosomes. That is why they called as transfer RNA. They transfer amino acid from cytoplasm to protein synthesizing machine to ribosomes. So primary structure of tRNA is important. It is single stranded because it is RNA single stranded with the 71 to 80 nucleotide long. So 3' end terminate with the sequence CCA. Secondary structure says each single stranded tRNA molecule fold up into clover leaf shaped structure. This is the one they folded and looks like clover leaf shaped structure having 4 main arms. This is exactly like not exactly, resembles like clover leaf shaped structure with 4 main arms: acceptor arm, okay, acceptor arm, anticodon arm, okay, down you can see arm D arm, and T/C arm. Okay, there are 4 arms: T/C arm. The folded structure is stabilized by hydrogen bonds between the complementary bases at different regions of same strand. So that is the structure actually. This is okay, this structure you can remember for the exam. T arm is it contains dihydrouracil, unusual base, okay, special base. So dihydrouracil is present. So this is actually helps in recognition of tRNA by the enzyme amino acyl tRNA synthase. T/C arm contains 2 unusual nucleotide, ribothymidine and pseudouridine. Right side you can see T/C loop contains ribothymidine and pseudouridine. They are nucleotides. Okay. So acceptor arm at the 3' end, acceptor arm at the 3' end of each tRNA has a base sequence CCA, okay, which takes up the amino acid, CCA, which takes up the amino acid, and anticodon arm opposite to the acceptor arm. Anticodon is down, acceptor is up. Acceptor anticodon opposite to acceptor arm has a loop of 3 unpaired bases called anticodon. Okay, the specificity of tRNA resides in the anticodon arm, which has base sequence complementary to that of mRNA codon. That is important. The amino acid corresponding to the anticodon comes and attaches to acceptor arm. Okay, so amino acid corresponding to the anticodon comes and attaches to the acceptor arm. So that is the specific thing. So this acceptor arm has base sequences complementary to mRNA codon. Okay, so as per the function of tRNA concerned, it serves as a adapter molecule to transfer a specific amino acid to the ribosome. Okay, specified by the codon on the mRNA for incorporation at a particular position of the protein molecule. So you have to remember structure of tRNA. Okay, clover leaf shaped structure with the 4 arms. Okay, this diagram is also better, and you have to write about pseudo uridine. Okay, uh, that unusual bases you have to write. Okay, ribothiamidine pseudouridine. Okay, and about 4 arms you have to write: acceptor arm, anticodon arm, D arm, and T/arm. And for DNA, deoxyribonucleic acid, choose this diagram, right one, not just ladder. Okay, twisted helix, and write the silent features also. Okay, thank you.